Hello, let's talk about plants. All right, so what plants did we look at through microscopes? Hopefully that's an easy question for you to answer. Go ahead and flip to your notebook plant section with your photographs and the chart of animal and plant cell. We're going to look together at some photographs that I pulled from student work that I thought were especially notable. So kudos, Jackson, Olivia, Kayla, and Levi. Those were pretty great photographs. Kayla and Levi's photos were of the skin of the onion bulb. Jackson's is of the root tip, onion root tip, and Olivia's is of the Elodea leaf. We're going to look at this list of cell structures and see what we can observe from our microscopic images and what we already know about them molecularly. So first stop is cell membrane. It's kind of hard to know if we're seeing the cell membrane or not at this level, because primarily what we're seeing dividing up these cells here and in the onion skin photographs and in the onion root tip photographs is primarily the cell wall. So what we're seeing is the cell wall. It shows up much more clearly, but we do know that it's just inside that cell wall. So we know it's there and we know something about it. So I'm going to mark it. I'm going to put a little circle around it. You can highlight it or underline it or something like that in your notes saying, wow, this is something we know something about. And what do we know about it? Well, of course, we know that it is made of a phospholipid bilayer. Temporary vacuole is an animal thing. We're going to ignore that for now. Next is nuclear membrane. As we know, eukaryotes are defined by having a membrane-bound nucleus. And you know what that membrane is made of? You got it. That membrane is made of a phospholipid bilayer. So lots of the structures we are learning about and will learn about have membranes around them or in them. That means they're made of a phospholipid bilayer. So that nuclear membrane, go ahead and pop a circle around it or highlight it somehow. We can see that not in the Elodea, but in the onion root tip, that's the boundary around these nuclei. All right, and DNA. Well, we actually can see DNA. That's the stuff, as you know, that's inside the nucleus. DNA is in the nucleus, but we can even see some kind of shape and structure to that DNA. If we look at these onion root tip cells, we notice that the nuclei don't all look alike, right? This one here doesn't even seem to have a membrane around it. It looks like someone dropped a pile of hot dogs on the floor. This one looks like a earthworm family trying to escape a house on fire. I don't know. Or they don't look the same as these other nuclei. Do you know why? Let's think about it. These cells are at the very end, the tip of an onion root. Now, if you're the root of a plant, you are constantly growing, right? You are growing deeper into the soil. So this is the tip of the root. That's where the most active growth is. For it to grow, what do you think these cells are undergoing? If you're thinking, oh my God, she's referring to mitosis, my favorite function in biology, you're right. They're doing mitosis. They're doing cell division. They're busy making more cells so that root tip can get deeper and deeper and deeper in the soil. So to divide, to make more cells, you've got to divide the contents of your nucleus super carefully to make sure each new cell has exact copies. And in that process, you chunk up your DNA into cute little X shapes like you've seen in 23andMe, and anytime you've seen a chromosome picture anywhere, that little cute little X shape. And that's what you're seeing here. These hot dogs on the floor are actually the plant's chromosomes. This onion plant's chromosomes have gotten kind of chunky and organized in the process of cell division. So each of these cells is kind of in a different stage of the cell cycle, but like this one down here and this one up here are very obviously in the process of dividing. And so they look very different. And when we look at that, we know we are looking at DNA. We're looking at DNA molecules. So we'll just put a little circle there on the DNA. And of course, we know DNA is made of nucleotides. Those nucleotides have 3 parts: a phosphate group, a sugar, and a nitrogenous base. And there are 4 different options: adenine, thymine, guanine, and cytosine. And we know that its job is to keep the code for the cell's proteins. Like if you're gonna have to make proteins, and the DNA holds the code for making proteins. So we know quite a bit about what is in this pile of hot dogs. Next, we have not talked about the nucleolus, and we're not really going to talk about it yet, but you can see it, right? Over here you see it's like a dark mass inside the nucleus. Hopefully in your pictures as well, you can not only see the different conditions of these nuclei, but some of them, like this one here, here, almost like a ghost-like dot. In a number of these, there's like a dark dot inside the nucleus, and that is the nucleolus. And we will learn about it later. But for now, we can be ecstatic to know that we could actually see that through a microscope. Of course, we saw the nucleus. We'll put a little circle around that. Woohoo! We're chugging. Okay, chloroplast. So we didn't see any chloroplasts in the root tip. Why do you think we didn't see any chloroplasts in the root tip? We only see the nucleus because it's been dyed. This root tip slide had a dye applied to it, which made some things show up better, especially the nucleus and the DNA. That's why we didn't see a nucleus in these cells or these cells or these cells. There was no dye. So you might be thinking, it's the dye, Miss Sanders. That's not a bad answer. But let me put it to you this way. Do you think the cells in the roots of a plant should have chloroplasts? Because hopefully you know, and if you don't, you know now, that chloroplasts are where photosynthesis happens. And surely you've learned in your life that photosynthesis is the process by which plants use solar energy to make their own food, right? So if the roots never see the light of day, there's no point in having chloroplasts in them. So they don't have them because they don't need them. There's no point. Why waste energy making them? However, in the leaf, and the only leaf we looked at was Elodia, we saw bunches of them. This is just an amazing picture. Olivia did an amazing job here. But hopefully you also saw little green dots inside your boxy cell structures, and those are the chloroplasts. And those chloroplasts are green because they have chlorophyll, a green pigment, to do photosynthesis. Those chloroplasts are green, and therefore plant structures like leaves are green because of the chlorophyll. All right, what else can we see? All the liquidy stuff is the cytoplasm. We saw cytoplasmic streaming in Elodia. Sometimes you could see that. We saw it in amoebas, the cytoplasm moving as the amoeba moved. Obviously, that takes energy, which is a characteristic of life. Endoplasmic reticulum, haven't talked about it, can't see it, leaving it alone. Ditto with ribosomes. Lysosome is in animals, not plants. Mitochondria, plants have them. We can't see them, leaving it alone. Vesicle. Go ahead and add a little circle structure and a line to vesicle, because plants actually do use vesicles. So the last thing in here that we kind of can see and know something about is the cell wall. Obviously, that's the easiest to observe of all of our pictures. We could see that for all of everything we looked at. We also know something about it for plants, right? So the cell wall we know is made with cellulose to be strong. So you're ready to answer some questions and move on.